Good afternoon, everybody. We've got our next chapter on DNA, deoxyribonucleic acid, and everything that goes along with it. So, get ready, and here we go. So let's start out with what DNA stands for. DNA, the D stands for deoxy, the N stands for nucleic, the A stands for acid. So the deoxy part means no oxygen. The ribo part refers to the sugar ribose, and then the N refers to nucleic acid, which just means that it's an acid of some sort. The job of DNA is to store and transmit genetic information, heredity. So what you pass on to your children, it's all stored in this. DNA is found in all cells, although if you remember the two main types of cells, you have eukaryotic and prokaryotic. In a eukaryote, DNA is found in the nucleus, and in a prokaryote, it's found in the area called the nucleoid, which is just a bundle of DNA with no nucleus around it. Okay, let's take a look at the structure of DNA. DNA is made up of two chains of nucleotides. Nucleotides are, if you remember, the monomer, or the building unit of nucleic acids. So they're nucleotides. So I'm going to circle one for you right here. There's a nucleotide. And the way that they arrange themselves, that they stack on top of each other like this. But the other side, in order to make them fit together here, they have to be upside down. So you see this guy's going this way, and this guy is going that way. So two chains of nucleotides that run in opposite directions. It's the only way that they will fit together here. Okay, we said that they're made out of nucleotides. Nucleotide is the building block, the Lego piece of DNA. These are the monomers in nucleic acids, and they have three parts. They have the sugar, deoxyribose, so this is the sugar. And since it's DNA, the sugar is deoxyribose, so ribose with no oxygen. We have a phosphate group, which is over here, just like ATP. If you remember, ATP was just this, but with three phosphates. And we have a base. And so right here, a base just means the opposite of an acid, and it can come in A, T, G, or C for DNA. For RNA, there's another one, uracil. We'll talk about that one later. So this is the basic building block upon which DNA is built. Of the bases, we said that there's four. A stands for adenine, T stands for thymine, C stands for cytosine, and G stands for guanine. Now the bases come in two varieties, 1 rings and 2 rings. The 2 ring guys are called purines, and the 1 ring guys are called pyrimidines. Now the pyrimidines always pair up with the purine. So A and T always go together, G and C always go together, because the width of DNA is going to be 3 rings wide. 1, 2, 3. 1, 2, 3. I can't put these two guys together because it's only 2 rings wide. And if I put these two guys together, that would be 4 rings wide. And I can't fit these two together, even though it would still be 3 rings wide. I can't because of the bonds that pop out of these corners right here won't work. So we'll explain that in a little bit. So purines are A, adenine and guanine. Pyrimidines are cytosine and thymine. The base pairing rules says who gets to bond to who. A always goes with T, G always goes with C. If you remember, I gave you the silly little thing at the beginning of the year. Ant and Tia, and cat and gato, to help you remember who goes with who. Now the reason why that they always go together in that way has to do with the structure of the base. So if we look at adenine, he's got this nitrogen right here and this hydrogen right here, who is looking to bond with this oxygen and this hydrogen. Okay, the other 1 ring doesn't have that. See, he's got 3 spots. 1, 2, 3. So this guy who has 2 binding sites can only bind to someone else with 2 binding sites, which is why A and T always go together. Now this one, we can see he's got 1, 2, 3 binding sites, and so does C. And that's why they always go together. So remember how I said that even though we have a 2 ring and a 1 ring, these two can't fit together because he's got 2 binding sites, he's got 3 binding sites, so they won't fit together. So that's why A and T always go together and G and C go together. So those binding sites, they form together right here, and they make what's called a hydrogen bond. We talked about hydrogen bonds before, that these bonds are like Velcro because they are easily ripped apart if needed. So you can see that A and T have 2 hydrogen bonds, G and C have 3 hydrogen bonds. How do I remember that? T for 2, G, C, and 3 all rhyme. So A and T, T for 2, G, C, 3, they all rhyme. Okay, that's how I can remember how many bonds are in between each one. So these bonds, because they're hydrogen, makes my DNA able to split in half quite easily. What I don't want to split is this direction. And so there we have much stronger bonds, covalent bonds, and they're holding together the sugars and the phosphates. Here's my sugars, and here's my phosphates. So we call this the backbone of DNA. So we say that the backbone of DNA is made up of alternating sugars and phosphates. The bases are in the middle, and the bases are directly attached to the sugar. So the phosphates are always just kind of in between here. Okay, so you can kind of go sugar, base, phosphate, sugar, base, phosphate, like that. And then you'll either have 1, 2, or 3 bonds to another base, sugar, phosphate, base, sugar, phosphate, like that. But they'd be upside down the other side. Okay, so hydrogen bonds here in the middle, covalent bonds here. DNA, when we look inside of a cell, if here's a cell, here's a nucleus. If I look in here to where the DNA is, we have 46 strands of it, and so to get tangled up. So DNA is normally either found as chromatin or chromosomes in a cell. Chromatin, if you remember, is just kind of like a big old hairball. Chromosomes are little furry X's, like this. Okay, we don't see 46 strands of DNA just lined up like that. So how do they turn into chromatin? What they do, the strand of DNA starts to wrap around these little balls of proteins called histones. A histone is a protein that DNA winds around. And then it winds around it a couple more times, like this, like that. So it makes what looks like a beaded necklace. And then pretty soon the histones all start to coil up on themselves and make a thicker rope. And these histones, what they do is when they join together, they make a little clump, actually 8 of them. They make a clump called a nucleosome. So we see DNA, and then we see 8 histones all packed together. DNA, 8 histones. And so now it looks like an even bigger beaded necklace. And so these nucleosomes, then they start sitting on top of each other like this, and that makes a big old rope. That thick rope is now able to just kind of coil up on itself and turn into chromatin, which is the loosely relaxed form of DNA. Even though it is tightly packed up, it's still considered loosely bound. Now when DNA is getting ready to when a cell's getting ready to divide, each strand of DNA needs to replicate itself. So here's one strand, and it's all coiled up on itself like this. And the DNA will copy itself and make an identical copy, and they stick together in the middle like this. And together we call this thing a chromosome. So a chromosome is what you find when a cell is about ready to divide. We only see that during mitosis. So the only time we see a chromosome is during mitosis. The rest of the time it's chromatin. So this is the more condensed version of DNA. And each half of the X is made out of something called a chromatid. So this is one chromatid, which is 1/2 of a replicated chromosome, which equals 1 strand of DNA. The other chromatid is the photocopy. of this strand of DNA. So here's your original, here's your photocopy. The two of them stick together, and that's what we call a chromosome. Okay? That's pretty much it for the basic structure of DNA and how it's formed inside of your cells. And so we're going to practice putting together some DNA strands, and then we'll move on to how you replicate DNA. All right, see you later, bye.